Hello and welcome to Problem Set 25. Our problems today will be from the topic nucleic acids. Before we get started, just a quick reminder that the worksheet to accompany this video is available online through my website. And with that said, let's get started with number one. Number one, what base and what sugar are in the following nucleosides and nucleotides? Okay, so what base and what sugar? Let's go to the blue pen. You'll see that I have A through D already written out on the board to make this go a little bit faster. So for A, we have deoxyadenosine. The deoxy prefix tells us that the sugar must be deoxyribose. So I'm going to write sugar equals deoxyribose. Adenosine tells us that the base is adenine. Remember that we changed the ending for the nucleoside name. So the name of the base is adenine. Go back to the original ending. Uridine. Okay, the lack of a prefix tells us that this nucleoside has the sugar ribose, and the base must be uracil. We know that this is a nucleoside because the ending has been changed for the pyrimidine to the IDINE ending, so we know it has a sugar. The lack of the prefix tells us the sugar is ribose. Deoxyguanosine. So the prefix gives the sugar away right away. The sugar in this nucleoside is deoxyribose. And the base must be guanine. Again, we changed the ending because it's a purine to OSINE. So when you name the base, make sure you go back to the original ending for the base and call it guanine. Guanosine is the name of a nucleoside. Okay. And last but not least, guanosine 5' monophosphate. So this is a nucleotide. The base and the sugar involved include the sugar ribose. I know that because it lacks the prefix deoxy. And the base is guanine. Again, the base name is not guanosine. Guanosine would be the name of a nucleoside. Okay, so be very careful on that. All right, going on to problem number 2. Label the following as a base, sugar, nucleoside, or nucleotide. Okay, so we're classifying for number 2, and I've got them written all up here on the board, again, to make this go a little bit faster. So for the first one, A, we have deoxyribose. That is simply the name of a sugar. So A is the name of a sugar. B, we have adenosine. Be very careful here. The ending of the name has changed to OSINE. That purine has had the ending change to reflect that it is a nucleoside. A nucleoside made up of adenine and the sugar ribose, which is why there's no prefix at the beginning of the name. So this is the name of a nucleoside. Going on to C, we have thymine. Notice that the ending of the name the ending of the base name has not been changed. This is a pyrimidine. If it were linked to a sugar, we would expect the ending of the name to be IDINE, thymidine. It has not been changed, telling us that this is simply the free base. It's not linked to a sugar or a phosphate or anything else. Deoxyguanosine. We know that the sugar is deoxyribose. We can see that it's linked to the base guanine, making this a nucleoside. Not a nucleotide, because a nucleotide has an extra third component, the phosphate. Cytidine. Now this one I think is confusing, okay, because the original base name is cytosine. And so a lot of people will see the name cytosine and see the O-S-I-N-E ending and think that that's a nucleoside. But cytosine is the name of just the free base. When we change the ending to I-D-I-N-E, because it is a pyrimidine, we know that we're talking about a cytosine, a nitrogen-containing base cytosine that has been linked to a sugar. In this case, there is no prefix, so it must be linked to the sugar ribose, making this a nucleoside. I'm going to add a little note over here. Note, be careful with cytosine, because the normal base name has the O-S-I-N-E ending, but it's not a purine. So remember that this is just the free base. It's a pyrimidine, so when it becomes a nucleoside, the ending changes to I-D-I-N-E, and that's how we know that cytidine is the nucleoside. Deoxythymidine 5' monophosphate. So the monophosphate probably gives away that this is a nucleotide pretty easily. But just to kind of break it down and make sure that we understand what this nucleotide looks like, notice the deoxy prefix, so this has a sugar, deoxyribose. Thymidine, so we know that the base is thymine. And monophosphate, so we know that this nucleotide has one phosphate. So there's your three components, your sugar, your base, and your phosphate, making this a nucleotide with a T, not an S. All right. Let's go ahead and do number 3. We have a little bit of room here before we go on. Number 3. Why is there a prime symbol after the 3 and the 5 when writing a DNA sequence? So remember that we need the 3' and the 5' because that prime tells us that we're counting within the ring of the sugar, the positions in the ring of the sugar, as opposed to the position of the rings in the nitrogen-containing bases. So let's write that here. The prime tells us that we are counting the positions within the sugar, not the base. Okay. All right, so we have definitely filled up our board. Let's go ahead and erase here before we go on to number 4. Number 4, what base is found only in RNA and what base is found only in DNA? So we know that in RNA, the base uracil is exclusively found. In DNA, it's thymine. And the reason for that is that when the RNA is copied from the DNA, remember it's just one gene copied from DNA, everywhere that a thymine occurs in the DNA, the RNA replaces it with a uracil. Okay, so the RNA is never going to have thymine because it's always going to replace thymines with a uracil when synthesized. Number 5. Which bases are found in both DNA and RNA? Okay, so found in both would be adenine, guanine, and cytosine. And number 6. How are nucleotides connected in a polynucleotide chain? Remember that the OH off of the 3' position of one nucleotide reacts with the phosphate off of the 5' position of another nucleotide, creating a phosphate diester linkage. So the short answer is they're connected by phosphate diester linkages. But let's go ahead and write down all that detail here. The OH of the 3' position of one nucleotide reacts with the phosphate off of the 5' position of another nucleotide, creating a phosphate diester linkage between the two nucleotides. Okay, so I'll underline phosphate diester linkage because that's what's really connecting them there. All right, number 7. What kind of intermolecular forces of attraction hold the DNA double helix together? Remember that that is hydrogen bonding. Specifically, the hydrogen bonding occurs between adenine and thymine and cytosine and guanine. So let me write here, A to T. Remember there's 2 hydrogen bonds between those 2 bases. And C to G. Remember there's 3 hydrogen bonds between those 2 bases. What we call complementary base pairing. All right, number 8. I think we can squeak it in here. How is 3' CAT 5' different from 5' 5' CAT 3'? Okay, so these are not the same, so I'm going to write not equal. These are not the same. Remember that the order of the nucleotides in the polynucleotide strand is important, it's significant. And we can see that the free OH, the 3' end, is going to be off of cytosine in the first polynucleotide here. In the second, the cytosine is not going to have a free OH because it's the 5' end. It's going to have a free phosphate. So although they're made up of the same 3 nucleotides, they're going to be different polynucleotides. And you can say the same thing about thymine. In the first, thymine is going to have a free phosphate because it's your 5' end. But in the second, thymine has a free OH because it's the 3' end. So let's write here that in the first polynucleotide, C has a free OH because it is at the 3' position, or end. Let's just write end, 3' end. But in the second polynucleotide. C has a free phosphate because it is at the 5' end. Okay, number 9, our final problem. In the double helix, what would be the complementary strand for the following? 5' ACA, TCT, GGG, CGT, ATA, CGT, 3'. So we know that the complementary strand, the strand that would run anti-parallel, would then have the 3' end here and the 5' end here. That's what is meant by anti-parallel. And then we just have to figure out the complementary base pairing that would be occurring. A, always complementary base pairs to T, C to G, A again to T, T complementary base pairs to A, C to G, T to A, G, 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 that's easy, C, 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 C to G, G to C, T to an A complementary base pairs to T, T to an A to a T, C to a G, G to a C, and T to an A. So this would be our complementary strand for this sequence here. Again, anti-parallel. Make sure you get the 3' and the 5' in the correct positions. All right, this will conclude our problem set 25. Thank you very much for tuning in, and I hope you will tune in to tutorial 26.